Hi, everyone. I will resist the urge to say good morning, and instead I will just say hello and welcome to the 6th Life Science Across the Globe seminar. My name is Janine Stevens. I am a Director of Scientific Programs at Janelia and the organizer of this seminar series. As always, I want to remind you that you can find more information on the series, including a full schedule of speakers and recordings of all past talks on our website at lifescienceacrosstheglobe.org. So last week we heard from EMBL in Europe, and today we are extremely excited to virtually travel to Africa, where it is significantly cooler today than it is here in Virginia. And we will highlight some science and some scientific culture from the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town in South Africa. Introducing our speakers today is Professor of Immunology and Chair and Head of the Division of Immunology at IDM, Clive Gray.
Well, I will say good morning, good evening, and good day to everyone. So it gives me great pleasure to introduce two of my colleagues from the University of Cape Town. Our first colleague is Professor Valerie Mizrahi, and my second colleague will be Anastasia Koch. So Valerie Mizrahi is the director of the Institute for Infectious Disease and Molecular Medicine here in Cape Town, and she also directs an extramural research unit at the South African Medical Research Council, which is also situated here in her unit in the IDM, and is a founding co-director of the National Center of Excellence in Biomedical TB Research. She was a senior international research scholar of the HHMI from 2012 to 2017. Now she has a page of numerous leadership awards, both national and international, and I won't read those out to you. But her current research focuses on aspects of the physiology and metabolism of Mycobacterium tuberculosis relevant to TB drug resistance, Mycobacterial resistance, and TB drug discovery. And I believe you'll be hearing of some of that today. So Valerie has trained more than 70 postgraduate students and postdocs, and some of whom are now in very prominent leadership positions, both here in South Africa and internationally. And she has mentored many, many early career research fellows. So without further ado, I introduce Professor Valerie Mizrahi.
Well, good morning, good afternoon, and good evening to everybody. It's an absolute thrill for me to be speaking to you today from Cape Town. I'm going to be talking to you today as a TB biologist around the problem of tuberculosis. I'd like to firstly start by thanking Ron Vale, and Janine and the whole team at Janelia for the huge honor of including the IDM in this series. And so before I get going on the TB part of my talk, just a couple of words firstly about the IDM. Our institute is the youngest, I believe, among the sister institutes in the seminar series. We were only established in 2004, and it was set up as an institute to bring people together to study diseases of importance on the African continent through research that we conduct at the interface of the laboratory, the clinic, and the community. As you'll hear from Anastasia, community engagement and public engagement is a core feature of the work that we do. The second core feature of the IDM is that we use research as the vehicle for training in Africa for Africa. So in other words, our research and our training enterprises are completely interlinked with one another. And as an African institution, we feel that this is critically important. The IDM is based at the University of Cape Town, which is a leading university on the African continent that is known for many areas of research strength, but of relevance today is the research on HIV, TB, and HIV-associated tuberculosis. And I've given the logos of 3 particularly well-known centers that fall under the umbrella of the IDM. To put today's talk in context, I'd like to share with you my personal perspective, and I hope that some of what I'm going to say on this slide resonates with colleagues, particularly elsewhere in Africa, in Latin America, and in Asia. I feel very strongly that as a disease-endemic country that has expertise and infrastructure, South Africa and other high-burden countries have an important role to play in the development of new approaches for tackling diseases such as TB. I also feel that scientists such as myself who live in disease and in high burden countries should and must engage in addressing fundamental questions in tuberculosis research. And again, this applies to other diseases that require high quality life sciences as well as health sciences research. I myself am a TB biologist. I'm actually a chemist by training, and I've spent many years in my career trying to understand the physiology and the metabolism of MTB, Mycobacterium tuberculosis, the causative agent of TB. And today I'm going to tell you about the way in which we've been applying insight into TB physiology and metabolism in drug discovery. So a few words first about TB. TB is today the leading infectious killer globally, worth bearing in mind in this time of COVID. Each and every year, 10 million people fall ill with this disease. About 1.5 million people die. A less known fact is that TB is a major cause as a result of antimicrobial resistance. About 1 in 4 AMR fatalities annually are attributable to tuberculosis. In my own country, the epidemic is a very serious one. We are one of the highest TB-burdened countries in the world. I won't read these statistics to you. You can see them yourselves. Suffice it to say that each and every day, 170 people approximately die of tuberculosis in South Africa. The majority are among people living with HIV. 60% of TB cases are HIV associated. And drug resistance has a disproportionate impact on this epidemic because it is difficult and costly to treat. So intervening in tuberculosis, TB research today really is geared towards intervening by developing new diagnostics, biomarkers, vaccines, and drugs. And these red squares denote the spaces in which these fields of research are aimed at. If one looks at the spectrum of individuals who are either infection eliminated or totally uninfected, which in disease endemic countries is a very small percentage, have not been exposed to the tubercle bacillus, all the way to an active TB patient. Today I'm going to focus entirely on drugs. You'll be hearing later in the series more about vaccine research from Tom Scriber. But today I'm going to talk about drugs which are aimed at going after active TB patients and improving the way that we can treat tuberculosis, which currently requires for drug-sensitive TB something called short-course chemotherapy, a contradiction in terms given that this takes a minimum of 6 months for drug-sensitive TB and a lot longer for drug-resistant TB. So what does the pipeline look like? A TB drug pipeline of this type with so many molecules in it would have been unimaginable 15 to 20 years ago, but in the post-genome era, thanks to public-private partnerships largely, there's been an explosion of interest, and we have a reasonably good pipeline today that has delivered these 3 drugs, which are really stand to revolutionize the treatment of drug-resistant tuberculosis and have gained market approval. The problem with pipelines is that risks go up and costs go up as one moves from the left to the right. And this is a resource-limited field, underscoring the need to populate the pipeline with the highest quality leads and new targets that ultimately can deliver new TB drugs that do 2 important things. The first is that they are active against drug-resistant strains of Mycobacterium tuberculosis, multi-drug resistant and extensively drug-resistant, as well as drug-sensitive TB, and achieve what is essentially an aspirational goal, dramatic treatment shortening. I'll come back to this topic later and touch on why I call it aspirational. In thinking about how to craft today's talk, I thought it would be useful to explain what we're trying to do as a complexity-simplicity paradox. What do I mean by that? It is fundamentally flawed to think about tuberculosis as a unitary disease. This is a disease that is fraught with biological as well as clinical complexity. Biological at the level of the pathogen, genetic and phenotypic heterogeneity. I'll touch a lot on phenotypic heterogeneity in this talk, as well as lesional genetic heterogeneity between hosts within hosts. So there's enormous amount of complexity. And yet what we aim to do is to develop simple interventions that are compatible with health systems in disease endemic countries. The fragility of health systems has certainly been brought into sharp relief in this time of COVID, not only in poorer countries, but certainly in well-resourced countries to the north. In the case of TB drugs, we're looking for short, safe, and effective drug regimens that can give rise to durable, sterilizing cure. And with respect, this presents major scientific challenges. So I became interested, having studied and worked on Mycobacterium tuberculosis biology for several years, and asking the question as to how we as biologists could use biological insight of MTB, learned from studies in vitro, in macrophages, in animal models of infection, and feed this into drug discovery by delivering validated drug targets, as well as bringing to bear the entire toolbox of omics technologies, imaging technologies to help to identify small molecules that can either go on and become drug candidates if you're lucky, but even if not, chemical probes are very, very useful for driving biological insight. So we drive the whole process in reverse, and this becomes mutually reinforcing. So to give context to the vignette that I want to share with you today, the first important message I want to convey is about the complexity of the encounters that MTB has during the course of transmission, infection, and disease. This rod-shaped bacillus, which is suspended in a bioaerosol, it gets inhaled into the lungs of an individual, encounters a very indifferent environment there than when it gets phagocytosed by phagocytic cells and then gets basically walled off in this cellular granuloma, which is the hallmark distinct feature of TB infection. In a subset of individuals, TB infection precedes to disease, and associated with this process is the breakdown of these granulomas from being cellular, highly organized granulomas that are well vascularized to so-called necrotic granulomas, which triggers the immune pathology that is associated with tuberculosis, which is lung damage, cavitation, and these bacteria can then escape out of these granulomas into the airways and go on and reinfect a new host. MTB is an organism that has 4,000 genes in its genome and is equipped with a very, very flexible, robust metabolism. So it stands to reason that in each of these environments, the bacterium adapts its physiology to give rise to a mixed population of bacilli that are sitting in a variety of metabolic states, and this greatly complicates treatment. Why do I say this? If one has a look at this caseating granuloma, these tubercle bacilli that are sitting in the caseum, which is at the center of this necrotic granuloma, are sitting in a poorly vascularized environment, is essentially hypoxic. The lipids that have been released from foam cells as they die and spill their contents into the caseum, is the carbon source comprises cholesterol esters as well as fatty acids. That's a very different environment than the bacteria sitting in macrophages or the bacteria that are extracellular. In the caseum, the TCA cycle is running in reverse, for example, whereas extracellular bacteria have no limitation to oxygen that may be sitting in the apex of the lung, for instance, enabling the bacillus to grow. This complicates treatment because a drug that acts on a cellular pathway that is necessary for bacterial growth might not deal with all the organisms. So understanding this kind of complexity is the type of thinking that biologists bring into the drug discovery space. Historically, in the post-genome era, our infatuation with identifying essential genes has triggered 10, 15 years ago a massive effort in so-called target-based approaches to drug discovery. When one picks your favorite essential target, develops a potent inhibitor against that target, and then hopes that that will become a drug candidate. In fact, this approach is met with very limited success. Not unique to TB, common in antimicrobial drug discovery, confounded by a lack of information on the vulnerability of a target. By that I mean, how far do you want to knock down the level of that target in order to result in growth inhibition? And confounded by issues of permeability and metabolism. Metabolism here, I mean the ability of the bacterium to biotransform xenobiotics. MTB has a robust system, very much like the human liver. And permeability speaks to this slide, which I felt compelled to show because it illustrates one of the wonders of nature, which is the mycobacterial cell envelope, which is an incredibly elaborate structure that I just want to highlight a couple of key features to for those of you who don't know much about the biology of TB. It is not a gram-negative organism. It has some features of it. There is the cell wall comprising the peptidoglycan layer, and what makes the cell wall quite intriguing is that the peptidoglycan layer is covalently attached to the inner membrane of the inner leaflet of the mycobacterial outer membrane, which comprises these very long chain fatty acids through a complex carbohydrate, which is shown here in green, known as arabinogalactan. So one has a very large polymer, which is a covalently attached polymer that is actually a wax at physiological conditions. And as a result of this complex structure, MTB envelope is often held up as essentially Fort Knox, as as a permeation barrier, and is posited as the default explanation for why molecules that act on a cytosolic target might not actually act on whole mycobacterial cells. This is held up as a hypothesis. Very seldom has this been demonstrated, and we must remember that complex natural products such as rifampicin do get into mycobacterium. So I put this up as an area of very active investigation. We need to understand much more about small molecule permeation in MTB. So given the target-based approaches have been problematical, the pipeline, in fact, has been populated by going back to the future and doing phenotypic screening. Screen against the whole organism, and the drug candidates and the drugs that have recently been approved have come from this route. This too is complicated, however, because elucidating modes of action can be challenging. I'll give you a case study from my lab in the next few slides that shows how we were able to do this quickly, but ran into some problems later. But we were very interested in borrowing from some of the work that was pioneered in Merck, aimed at bringing together phenotypic and target-led approaches for drug discovery that take advantage of our ability to generate hypomorphs. These are conditional knockdown mutants in which one depletes the level of the target of interest, which enables one to do so-called target-based or target-biased whole cell screens, gearing your discovery of whole cell actives towards targets of pathways of interest. And we published on this one of the first studies, and it was exciting to see a group led by Deb Hung at the Broad Institute took this approach to scale and published this in Nature of last year. So I want to now share with you a case study from my own lab. It's the case of Ampdh, and the reason for the ambiguity in the title of the slide will become evident in a few minutes. The story has its origins in More Medicines for Tuberculosis, an EU-funded consortium that my lab belonged to, led by Stuart Cole, who was then in Switzerland at EPFL and is now president of Institut Pasteur in Paris. We were sent this compound that was generated by colleagues in Hungary, and it is an isoquinoline sulfonamide, and we were asked to elucidate the MOA, and Vinayak Singh, a postdoc in my lab, took the compound and showed very quickly it has a decent MIC, it is bactericidal, it shows time and concentration dependent kill of MTB, and Vin went and raised resistant mutants. They emerged at a low frequency in M. tuberculosis, and we found by whole genome sequencing an intriguing non-synonymous SNP that mapped to a tyrosine residue in a conserved region of a gene encoding IMPDH, inosine monophosphate dehydrogenase. Those of you who studied biochemistry, this is Biochemistry 101, involved in purine, Denovo purine biosynthesis. We thought that this might be the target, and our collaborators from MM4TB in Italy, Menico Rizzi's group, expressed the protein and showed indeed that this compound is a potent inhibitor. It has an uncompetitive mechanism of inhibition, potent inhibitor of the MTB enzyme, and Sir Tom Blundell's group at Cambridge co-crystallized the inhibitor with a substrate and showed very beautifully that the that the inhibitor shown in orange binds at the active site, and you can see the pi interaction with the isoquinoline of the compound and the hyperxanthine of IMP forms one pi stacking interaction. The second is between the cyclohexyl ring and the tyrosine 487. This is the tyrosine which is mutated to a cysteine. So very clearly we could see the loss of that tyrosine collapses the binding of the compound and you get a resistant mutant. To close the loop and to show that this compound in fact acts on IMPDH and MTB, Vinaya created a hypomorph, a knockdown mutant, where we dial down the level of IMPDH using a tetracycline inducer and showed potent hypersensitization. So we'd closed the loop completely, going from a phenotypic hit to a target to an inhibitor, and got excited about the target because of where it sits. So to orient you, this is the Denovo purine biosynthesis pathway. This is where IMPDH sits. It catalyzes the first committed step in the production of guanine nucleotides, and guanine nucleotide disruption will block not only nucleic acid biosynthesis, but GTP is required for multiple biochemical pathways. So great, we figured that we could detonate a bomb inside MTB with pleiotropic consequences. The problem with when you deal with a bug that not only packs its own lunch by producing its own metabolites, it can also salvage, it can steal from the host. And if guanine is available, the function of HMP could subvert the essentiality of IMPDH by going straight to guanosine monophosphate. So we needed to ask three questions. Does the salvage activity work? If so, how much guanine is needed? 3, does it matter in an in vivo infection? So we could answer the two questions easily by demonstrating that guanine supplementation rescues MTB from the inhibitory effects of the drug, as well as the inhibitory effect of IMPDH depletion, but the concentration is high. We need to go to above 100 micromolar guanine in the extracellular milieu in order to get this rescue to occur. Does MTB see this amount of guanine in an animal infection? Before I get into the animal infection, I want to show you these time-lapse movies for a number of reasons. Firstly, they're really cool. You can see the polar growth of MTB. This is the hypomorph in a time-lapse video that was produced by Niraj Dar at EPFL. The hypomorphs are growing in these microcolonies. And what happened at 72 hours, 3 days, we added tetracycline, which is the inducer of the knockdown system. So this results in transcriptional silencing of IMPDH, and you can see growth inhibition occurs. We don't see change in morphology of these bacteria, but upon prolonged silencing of IMPDH, you can see some of the bacteria start to die. They disappear. So this proves that this is indeed a bactericidal drug target, but look at the time. We are running into days of silencing. At 330 hours, we're going to do something different in the microfluidic device. We're going to add guanine and see whether the salvage pathway can trigger any regrowth. And what you can see in the top microfluidic, the top microcolony, complete sterilization. But look at what's happening at the bottom. You can see that there is regrowth, even though we suppressed the expression of an essential enzyme for 10 days. There was a lurking persister cell that was sitting in that microcolony that was able to sense, transport, and assimilate guanine, so it remained metabolically active. I show you this for various reasons. Firstly, it's a very visceral illustration of the heterogeneity of MTB cell death, the slowness of MTB cell death, and the persister problem of MTB. And it's relevant to the question that I'll touch on about treatment shortening. What happens now in an animal model of infection? Well, if we infect mice with the hypomorph and don't feed them any inducer of the knockdown system, you get growth and persistence like a typical virulent MTB. But if you feed these animals with doxycycline, what you find is rapid elimination of the bacteria to the point at which the lungs of those infected animals look as if they never ever saw TB. So clearly in the mouse model of infection, there's not enough guanine to rescue MTB from the inhibitory effects of IMPDH loss. So we called this a vulnerable essential target that had been validated genetically in mice and got excited about this target, but the reason for the ambiguity is that there was a second study happening in parallel to ours, led by our collaborators at NIH, Cliff Barry and Helena Boshoff, working with Paul Wyatt's group at Dundee, had discovered another series of compounds in dissolved sulfonamides hit exactly the same targets, but they called this a not vulnerable target. And the key was that they went and had a look in the tissue of different animals, as well as humans, at guanine levels. And what is illustrated on this slide are data from their publication, which show that guanine levels in the organs of mice are well below 50 micromolar, irrespective of the organ one's looking at. Whereas in fact, if one has a look at lung tissue from human individuals, TB patients who have undergone lung resections, or rabbits infected with tuberculosis, you can see that the scale is very different. Guanine levels sit above 500 micromolar, in fact, run into the millimolar levels. So quite clearly, this put a very big question mark over IMPDH as a target for drugs for human tuberculosis by virtue of the fact that the risk exists of salvage, bypassing the essentiality of the target. And the learning lesson here for me is the critical importance of understanding the metabolism of MTB in humans who we want to treat. Animal models are very valuable, but in this particular case, the variable environments which I've indicated previously, whether one sits in cellular versus necrotic granuloma, are likely to influence metabolic vulnerabilities, and we need to understand perfectly the metabolism of M. tuberculosis in humans. In the last two slides, as I come to an end now of my talk, I want to just come back to the question of the aspirational goal of treatment shortening and share with you what I consider to be an important reality check from the work of Richard White's group at the London School of Hygiene and Tropical Medicine. It's a modeling study asking the question of what a 4-month non-inferior regimen for first-line treatment of TB would do, and it would have, sadly, a small impact on deaths and alleys averted, but in fact it could be cost effective, so it is not to be dismissed. Remembering that current short-line regimens last for 6 months for drug-sensitive tuberculosis. So the question is, what is it going to take to achieve dramatic treatment shortening? And working in Cape Town, which is a premier site for testing new drugs and drug combinations that I showed you on the pipeline earlier, I am often very swayed looking at lessons from the clinic. This happens to be a lesson from an early bactericidal study done on a novel regimen at that time, preterminate, moxifloxacin and pyrazinamide, asking the question of how rapidly does the bacterial load in the lungs of a TB patient decline as a function of time on therapy. Shown in turquoise triangles is HRZE, the standard of care, the 4-drug regimen for tuberculosis. And you can see that the time at which culture conversion occurs, in other words, you intersect that dashed line and you can no longer cultivate bacteria, is between 7 and 8 weeks. And what is shown importantly in green is the high-dose preterminate arm of the clinical trial gave you the intersection at an earlier time, about 2 weeks earlier. This was met with great excitement because it was one of the first results showing in humans that new combinations can be treatment shortening. But as a microbiologist who's just shown you a time-lapse movie that shows you a slow and agonizing death of tuberculosis, of MTB, the question that we have to ask is, how far can we bend the kill curve? And I just want to leave you with what I consider to be some key approaches that groups are taking, coupled with some key questions. What will it take to achieve durable sterilizing cure that is dramatically shortened in treatment? Durable meaning it lasts, at least until the next infection. We know that people can and do get reinfected with TB. And sterilizing means killing every single bacillus that is lurking in that individual. In other words, the only good bacillus is a dead bacillus. So from a microbiological perspective, the approach that my lab and many others are taking is to ask the question of whether we can kill faster. We are looking at ways of trying to target factors that mitigate drug efficacy and thinking about bringing that kind of scientific knowledge into better ways of combining targets and drugs. If you're interested in this topic, I'll refer you to some beautiful work that Bree Ulrich at Tufts University is doing. The publications are cited below. And ultimately, as I've shown you, Bacilli sitting caseum are difficult to get to, and they are very slowly replicating. We need to understand the factors essential for survival in those environments, and then work with pharmacologists and chemists to build molecules that can get there. An entirely orthogonal approach, which is actually a big area of research in the IDM as well, is looking at host-directed therapies. And this orthogonal approach has strong parallels in COVID, antiretrovirals and host-directed therapies. In TB, host-directed therapies are designed to potentiate the efficacy of TB drugs. So in closing, to me, the messages I want to convey today is that as a biologist trying to work in driving drug discovery and informing drug discovery, I've learned that building clinical insight is critically important. And I do think that this three-way approach is very powerful for the future and offers great prospects for our ability to tackle this very, very nasty pathogen that gives rise to the world's leading cause of infectious disease. I'm going to stop there. I've thanked the people along the way during the talk. This is a gallery taken from last year of the group that I run together with my colleague, B. Warner. Over the years, I've had fantastic people working in the group. This group is just as good as any other group in the MMRU in the past. We're embedded in the IDM in Cape Town. We collaborate extensively internationally, and none of our work would have been possible without the great inspiration we get from colleagues across the world, as well as the many funders. And today I do want to especially acknowledge the HHMI, because being a basic scientist and being inspired as being part of the HHMI community as an international scholar encouraged me to do the kind of work that I've given you a little bit of insight into today. So I'm going to stop there and stop sharing my screen now. Thanks. Happy to take questions.
Thank you, Val. That was a fantastic, very interesting talk. We do have a lot of great questions. I'm going to start with this one. The isoquinoline sulfonamide compound. Can you tell us a little bit more about where that came from? Was that a synthetic drug library? And if so, are there other possibilities within that library being pursued?
Okay, so thanks for that question. It was a compound that was generated at Vichem, who are our collaborators in Hungary. It was a part of quite a small series of compounds. In fact, they had been generated as host kinase inhibitors, and phenotypic screening done at the EPFL took compounds irrespective of what they'd been designed for, and screened them against tuberculosis. So a host kinase inhibitor actually turns out to inhibit Ampdh. We subsequently published a paper about 6 months ago in European Journal of Medicinal Chemistry doing an SAR study looking at other analogs in that series. We were unable in that series to come up with better inhibitors than the one we had. We did interestingly find some that were active against the resistant form of Ampdh, but I know that there are several other questions, so I'm going to stop there. I'd be happy to give you further information if you contact me after this the call. Next question.
Thanks, sort of a follow-up question. Are there any other mycobacterial diseases that you're aware of that may be successfully treated by this drug? For example, ones that are non-lung related?
Yeah, no, I think, again, that's a great question, and it gives me a segue into saying that one of the big pluses of this quite coordinated approach to TB drug discovery, the spin-off consequences are in other mycobacterial diseases. If TB is under-resourced, then other diseases such as leprosy and borreliosis, for example, are chronically under-resourced and fall in the category of truly neglected diseases. Ampdh, it's a good question as to whether leprosy has a homolog. I should know that, but it's not clear in my head at this point in time. Potentially for others, the abscesses, for example. But rather than focus on Ampdh, what I should mention is another really interesting area of research that my lab worked on for many years, which was looking at energy metabolism. And that it turns out to be a very hot area of drug discovery. And there's some beautiful work that Kevin Pett and others in Singapore have done to show that inhibitors that go after an enzyme involved in energy energy metabolism. It's part of the respiratory chain of M. tuberculosis that are quite good in MTB are actually very, very good against other mycobacterial diseases. So again, what I would encourage people who are working outside on mycobacterial diseases outside of TB, we really need to join forces and see what learnings have come from TB. You can shortcut and short circuit all of this research work and take some of these hit compounds and test them against other non-tuberculous mycobacteria. This, in fact, is quite a well-funded area that the NIH is investing in non-tuberculous mycobacteria, such as Mycobacterium abscessus and others. But great question. Thank you.
Thanks, Val. We have a question now from Leopold, and I was wondering, Leopold, are you there? Would you like to ask your question?
Yeah, hello.
Yes, go ahead. Please ask your question.
Yeah, thank you for giving the floor, and really great presentation by Valerie that I know very well. I was very interested in your presentation. I was just wondering, with the development of the molecular method, and now we know more and more how complex the mycobacterial genus is, with different strains causing disease in different parts of the world. I was just wondering, how do you integrate the fact that mycobacteria is so different in your drug discovery initiative?
I think Leopold, that's a fantastic question. And of course, we concern ourselves very deeply with that question. Just for those who are not familiar, there are 7 lineages of M. tuberculosis. The more we're looking, the more diversity that we're finding. Obviously, the first question one asks when you find a drug target and look at a drug target is, what is the extent of variation within that target, and can that confer intrinsic resistance to the drug? And so typically drug discovery consortia will take representatives of the major lineages for counter screening purposes, but also importantly to counter screen against resistant isolates. Because again, a lesser known fact about TB is drug resistance is all mediated on the chromosome. There's no horizontal determinants of drug resistance. So Leopold, the short answer is that a lot of counter screening against representative strains is critically important, because as part of the de-risking process before one takes a compound forward, I should say this strain variation is a relative term. Relative to other bacterial pathogens, MTB still is relatively highly conserved and shows relatively little variation. And it hasn't, in my experience, popped up as being a major confounder because there's such high degree of conservation of essential cellular functions, but it is something that we have to be alert to.
If I can follow up.
Yeah, go ahead.
I mean, just to say a really great answer. I'm just saying, because this information is quite new, as you mentioned, since we have been really looking at very recently, I think more research needs to be done because it's not only chromosome, point mutation that drive resistance, but also very phenotypic. Yeah.
Absolutely. So it's a great question.
Let's see, Val, I think we have one other question here. There's not a name associated with it, so I'm going to just go ahead and read it. But this person would like to know, is there a way to activate bacteria inside of a granuloma so that it could become susceptible to more conventional inhibitors or bactericidal compounds?
Yeah, so I mean, that's a fantastic question. And nature sometimes does this. We reactivate, for instance, if one has HIV during pregnancy, immune suppression results in change of environment to the extent that the bacteria are able to now kind of move out of this very slow, so-called states of non-replicating persistence and start growing again. I think in answer to the question, host-directed therapy is geared towards doing exactly this, changing the environment, reducing the pathology that is the driver, we think, of creating these hostile environments that put MTB into the kind of hunker down mode, and creating environments that are more amenable to growth, because if you're more amenable to growth, you're going to be more amenable to death by antibiotics that target processes that are required for growth of the organism. So this really speaks to the orthogonal approach of host-directed therapy, but again, an intriguing thought and good question.
Thanks, Val. I think at this point we are going to move on to our second speaker.